Good morning, student. Now let's see today our next topic, that is the DNA replication. Okay, DNA replication, and this process of replication require a set of catalyzed enzymes. Okay, now these what are these catalyzed enzyme? First is the helicase. Okay, helicase, topoisomerase, primase, DNA polymerase, DNA ligase, and repair enzymes. These are the enzymes that are required for the DNA replication. Okay, so first is the main enzyme is the DNA polymerase. DNA polymerase enzyme is the main enzyme. You can say it is the main enzyme which uses a DNA template to catalyze the polymerization of deoxynucleotide. Okay, and the average rate of polymerization is 2000 base pair, okay, per second approximately. It's okay. And in prokaryote there are three types of DNA or DNA polymerase are found. Okay, and in the eukaryotes five different DNA polymerase are found. Okay, and these enzymes are highly efficient as they have to catalyze polymerization of a number of nucleotides in a very short time with a high degree of accuracy. Okay, as any mistake would result in the mutation. अगर mistake हो गई को polymerize करना होता है, अगर कोई mistake हो गया तो क्या हो जाएगा, mutation होता है. Okay, therefore this enzyme help in removing mismatched nucleotide by mechanism कॉल प्रूफ रीडिंग. ओके, तो एक मैकेनिज्म होता है प्रूफ रीडिंग, उसके अकॉर्डिंग टू दैट ये क्या करते हैं, हेल्प इन रिमूव मिसमैच नहीं हो, किसी भी कंडीशन में क्या नहीं हो, मिसमैच नहीं हो. ओके, न्यूक्लियोटाइड्स, गेट इट. ओके, तो दिस इज द डीएनए पॉलीमरेज. देन नेक्स्ट इज द हेलिकेज एंजाइम. हेलिकेज एंजाइम, दिस हेलिकेज एंजाइम क्या करता है ये, व्हाट इज द फंक्शन ऑफ इट, इट अनवाइंड द डीएनए स्ट्रैंड. ओके, अनवाइंड करेगा डीएनए स्ट्रैंड को, सेपरेट द टू स्ट्रैंड्स, separate the two strand from the one point for the formation of replication fork. जो replication fork बनेगा उसके लिए separate करेगा strands को. Okay, next is the topoisomerase. Topoisomerase, the unwinding of DNA create a tension in the DNA strand. Okay, unwind हुआ DNA तो वो एक tension create करता है DNA strand में, get it, and which get released by the enzyme. Okay, and it get released by the enzyme topoisomerase. Okay, ये क्या करेगा, क्रिएट द टेंशन इन द डीएनए व्हेन द डीएनए अनवाइंडिंग. ओके, द नेक्स्ट इज द डीएनए लाइगेज. ओके, डीएनए लाइगेज, इट फैसिलिटेट द जॉइनिंग. ओके, इट फैसिलिटेट द joining. DNA ligase, it facilitates the joining of DNA strand together by catalyzing the formation of phosphodiester bond. It play a role in pairing single strand break in duplex DNA. Ligase क्या करेगा, joining. ओके, फैसिलिटेट करेगा किसको, ज्वाइनिंग ऑफ डीएनए स्ट्रैंड. गेट इट, टुगेदर बाय कैटेलाइज करता है किसके फॉर्मेशन को, फॉस्फोडाइएस्टर बॉन्ड. ओके, एंड इट प्लेज रोल इन द रिपेयरिंग सिंगल स्ट्रैंड ऑल्सो. ओके, दैट इज द डीएनए ligase enzyme. So these are the enzyme that help in the DNA replication. Okay, get it. Now what are the steps process, you can say in the DNA replication, what are the steps. First is the you can say, okay, origination or organization of replication, get it. Second is unwinding of DNA molecule. Third is formation of RNA primer. Next is synthesis of complementary strand, then proofreading and DNA repair. Okay, so these are the, what are these, these are the steps of the DNA replication. Get it. Okay, अब now first origin of replication. First, origination of replication, what is it. Okay, replication, you know that replication start at a small opening of DNA helix, as a complete DNA cannot be separate. complete DNA separate नहीं होता है ना, एक छोटा सा opening जो DNA helix है वहाँ के start कहाँ से होगा, एक छोटे से opening से होता है. Okay, then initiation points or origin. हाइड्रोजन बॉन्ड क्या होंगे, between two bases breaks. Okay, हाइड्रोजन बॉन्ड टू बेसिस के बीच में क्या होगा, हाइड्रोजन बॉन्ड ब्रेक डाउन होते हैं. ओके, देन आफ्टर अनवाइंडिंग ऑफ डीएनए मॉलिक्यूल, अब ओरिजिनेशन हो गया, रेप्लिकेशन हो गया. ठीक है, डीएनए हेलिक्स के एक स्मॉल ओपनिंग से वो क्या स्टार्ट होंगे, सेपरेट होना स्टार्ट होंगे. फिर क्या होगा, अनवाइंडिंग होगी डीएनए मॉलिक्यूल्स की. ओके, अनवाइंडिंग होगा, अनवाइंडिंग ऑफ डीएनए मॉलिक्यूल्स. ओके, अब अनवाइंडिंग करने में क्या होगा, एंजाइम हेलिकेज एंजाइम क्या करेगा, हेल्प करेगा, दोनों डीएनए स्ट्रैंड को क्या करेगा, वो सेपरेट करेगा. ओके, देन weak hydrogen bond involving breaking weak hydrogen bond. For this now energy is required. Because replication is an energy expensive process. Okay, we can say replication is an energy expensive process. It requires the energy. Okay, get it? So now deoxyribonucleoside triphosphate D and TPS serve as the dual purpose. Okay, serve as your dual purpose of acting as a substrate and providing energy. वो यहाँ पर क्या करेगा? ATP जो है, okay, किस फॉर्म में होगी वो? deoxyribonucleoside triphosphate D and TPS and serve as a dual purpose, acting as a substrate की तरह वो act भी करेगी और energy को provide भी करेगी from two terminal phosphate. Okay, get it? Then enzyme topoisomerase cut and reseal one strand of DNA strand, helping in separation of intertwined strand of a long DNA molecule. Okay, and it leads to the formation of Y-shaped replication fork. क्या बनेगा? एक Y-shape का replication fork का क्या होगा? Formation होगा. Okay, then formation of RNA primer, short segment of RNA nucleotide. अब क्या बना आपका? Short segment बना RNA nucleotides formed at DNA template from the replication begins. Okay, initiation, the formation of new complementary DNA strand 3' and okay, get it? अब after उसके क्या होगा? In a long DNA molecule, replication take हो गया with a small opening of the DNA helix known as replication fork. उसको क्या बोला जाएगा? Replication fork बोला जाएगा. Okay, क्योंकि fork की तरह shape होता है उसका. Okay, and DNA dependent DNA polymerase catalyze polymerization only in one direction, 5' to 3'. Okay, this bring additional complications at the replication fork. Okay, अब क्या होगा? Due to this one strand template with polarity 3' to 5', replication is continuous and the DNA strand is called leading strand. उसको क्या बोलेंगे हम? DNA strand को उसको DNA strand को क्या बोला जाएगा? Leading strand बोला जाएगा. Okay, then this fragment synthesize the later joined by the enzyme DNA ligase. Okay, and the DNA polymerase cannot initiate replication process on their own. Also, replication does not initiate randomly where in DNA. Okay, get it? It began at a definite region in E. coli DNA molecule known as origin of replication. I already explained then, which has recognition site for the DNA. Okay, it begin at a definite region in polymerase replication हो गया origin which is recognized site for the DNA polymerase and which also provide the site for attachment of RNA primer. Okay, get it? And due to this requirement of the origin of replication, if a piece of DNA needs to be propagated during recombination, a vector is required and the vector provide the origin of replication. Get it? And in eukaryote, the replication of DNA take place at the S phase of the cell cycle. After DNA replication, if cell division fails to take place, it results in polyploidy or chromosomal anomaly. Okay, get it? So that is the DNA replication.